Good morning, good afternoon, and good evening to you all. I'm Anne Gourmelon, and I'm Principal Administrator of the OECD Test Guidelines Programme. My presentation today will provide you with some basic information about the functioning of the OECD Test Guidelines Programme and everything you ever wanted to know without daring to ask. So the foundation of the OECD Test Guidelines Programme are in the system of mutual acceptance of data, where a study following an OECD test guideline in accordance with good laboratory practice generate data that are accepted in all countries that have the same data requirement. This is further elaborated in details in an OECD Council decision dating from 1981. So tested once, accepted everywhere is the motto. The working party of the national coordinators of the test guidelines program oversees the work in the program. Members and adhering countries nominate one or more national coordinators. The European Commission, industry, non-governmental organizations such as animal welfare groups also have their national coordinators. The list is public, as you can see, and the secretariat team supports the program. The WNT, which is the acronym for this working party, meets once a year in April for about 4 days. Documents are submitted for approval on this occasion only. Decisions are made on project proposals. Proposals that are made by the member countries. The meeting is, of course, an occasion to review ongoing projects and discuss any issues encountered and also general program-related issues. Regular observers are the People's Republic of China and the Russian Federation. 2 advisory groups are affiliated to the program. The Endocrine Disruptor Testing and Assessment Advisory Group and the group on molecular screening and toxicogenomics that also oversees the Adverse Outcome Pathway Program at the OECD. And deliverables from this program, the ECMUST, are both relevant to the WNT and to another working party, which is the Working Party on Hazard Assessment. So projects in the test guideline program are led by member countries, as I already mentioned. Contributions to the program represent voluntary work by experts in member countries. The projects respond to specific regulatory needs in several countries. A template format for making proposals is available to national coordinators for describing new project proposals. And every year in November, proposals are invited and there is a review cycle that starts. looking a little bit more into the details of the template for making project proposals. Information is expected on the regulatory needs and the validation status of a proposed method, an endpoint, or an approach. The presence of protected elements has also become an important information indicative of the future accessibility, the transparency and distribution of new technologies covered in new approaches. So the review of proposals for new projects takes place between November and mid-January of the year after. So a couple of weeks, a few weeks, to organize some iterations of review and responses and revisions to project proposals until the annual meeting of the WNT takes a decision on a new project. On this slide, it shows you a snapshot of the different projects on the work plan in each area of chemical safety testing. And of course, you immediately see that half of the resources in the program are devoted to methodologies for human health, safety testing and assessment. The program is really rich of a vast network of experts nominated by their national coordinators. In expert groups, scientists from academia, regulatory agencies, national institutes, industry, contract research organizations, animal welfare groups, and other NGOs interact together to develop, review, and harmonize test guidelines, guidance documents, and review papers. Here on this slide, I indicated the expert groups that are currently in active discussions on the projects. So although the main objective of the program is to deliver the harmonized testing tools that can be used to meet the regulatory needs in member countries, the program also generates a number of companion documents and material to support test guidelines use, and review papers that are exploratory for new areas of testing and assessment. In the course of the development of a harmonized test guideline, there are several important steps that are ultimately leading to the acceptance of test guidelines. Typically, a test method that is candidate for test guideline development comes with a package of supporting information and data on its validation status to attest of its reproducibility and relevance. Guidance has been developed over the years to orient test method developers in the different steps that can be organized in modular approaches towards the development and validation of test methods. And later in this webinar, you will have presentation from Valérie Zouang from the Joint Research Center at the European Commission, who will further elaborate in the next talk on the validation in particular. So obviously, once a method is considered valid by the WNT and approved as a test guideline, subsequent demonstrations of its validity are not needed each time the test method is used, of course. This diagram summarizes the processes, the workflow, and the timelines for the test guideline development. So once a draft test guideline is ready, the draft test guideline is typically made available on the OECD public website, and commenting rounds are organized with the national coordinators to collect feedback and consolidate the test guideline. And obviously, the expert groups are playing a very important role at that stage to help the lead country for the project addressing the comments received. And the WNT then looks for a consensus on the final document once it is ready for approval. Then on this last slide, you have an overview of the test guidelines team at the OECD Secretariat and their respective areas of coverage for projects that are active at the moment. So with that, I thank you very much for your attention. I hope it was useful, and I will now hand over to the chair of the test guidelines, the working party of the national coordinators of the test guidelines program, Tim Singer from Canada, to say a few words from his perspective and his experience as chair. Tim, over to you.
Thank you so much, Anne, and welcome everyone, and thank you for joining. Certainly my pleasure today to share a few of my own personal reflections on the test guidelines program. Before going any further, I'll just pause quickly if, Anne, you could just advise that you can hear me.
Yes, we can hear you.
Thank you. So as I said, some personal reflections from me and my experience in the test guidelines program. It's actually been about 20 years, as hard as that is to believe, that I've had some involvement with the test guidelines program, be it as a user of test guidelines, someone who's authored some test guidelines and revised test guidelines, and then as a national coordinator for Canada, and then subsequently as the chair for the last number of years. So if we move forward in the first slide, as Anne indicated, the working party is in fact the governance body. And as all or most of the entities within the OECD operates on a consensus basis, as Anne indicated, with decisions made following an extensive consultative process. And so the diagram that I show here, that's essentially from Anne's presentation, indicates the extent to which these consultations occur. And decisions are extremely well considered before they're made. The downside, of course, is that this is a process that does take some time. And so you'll see that following innovations in methodologies that have applicability in the regulatory space, they're not immediately incorporated into the program. There is a process by which due diligence is done and experts are engaged before we see these approaches move into the test guidelines themselves. The role of national coordinators is actually somewhat of a challenging job at times for most of us as the focal points for the test guidelines program in our respective countries. because not only do we have the responsibilities to organize within our countries the efforts in support of the program and to engage with the program, which often means engaging with a fairly wide and large expert group community to organize the sharing of comments and perspectives on the work. But also we, as indicated, meet once a year for a meeting at which we represent our countries. The important thing to note about the body itself is that there remains from year to year a fairly consistent core group of national coordinators, with the turnover being relatively small. And so that means as a body corporate, we actually have a quite considerable corporate memory. And so as a result, we tend to make decisions with a degree of internal consistency that's quite high. And that means that decisions that are made are well considered, and they reflect the past precedents while incorporating changes that reflect advances in the science and the technology that warrants consideration. If we move forward, and I'll just start here with a few reflections on the past, and then I'll move forward in a moment to talk about where I think things might go for the future. But of course, over the last, I think it's been 40 years since the Council decision, obviously these tools have been well used amongst all member countries. So there's an enormous amount of experience that's been gained to date with test guidelines and the associated guidance documents that are developed to support them. And really the mutual acceptance of data concept or the principle has become really indispensable to regulatory organizations and industry worldwide, and not only avoiding duplication, but also being able to establish scientifically sound methods with the aim of protecting human health and the environment. And so these methods, having been extremely well essentially developed in the scientific capacity, then support regulation worldwide, and including in countries that are adherent and not necessarily members of the OECD, but use the test guidelines to support their own national regulatory regimes. I couldn't reflect on the past without noting the extent to which animal welfare considerations have really become fundamental to the program over the last 10 plus years. Although the program is really focused on addressing regulatory needs, it's not doing that without a strong focus on moving away from animal-based methods wherever possible, and to accept and embrace alternative methods. It's also worth noting, and you can see that actually if you put physically a test guideline written 20 years ago against a test guideline written today, just on a table, you can see definitely it's thicker. It's much more complicated and obviously much more lengthy, with the aim, I think, of trying to reduce ambiguities and to provide more clarity. There's a lot more content within test guidelines today than there's been in the past. And so from the perspective of those users of the test guidelines, the intention, of course, is to be more supportive, but of course, it comes with the cost of complexity at times. And then there's been some, and it's been more recent, I think, in my experience, efforts made to address issues associated with intellectual property and to avoid, I guess, what we call inadvertent monopolies. And so performance-based test guidelines, so these are test guidelines where there's standards essentially embedded within the test guideline to allow others to develop similar methods than those that incorporate some proprietary elements. And as a result of that, there's been in some areas for some endpoints an explosion of what we call Me Too, or very similar methodologies. So you'll see some endpoints, some areas in which the test guidelines cover being fairly well subscribed by various test guidelines, where there's, in some cases, less subscription of other methodologies. And then moving forward, talking about where I see things might go in the future. Obviously, Anne indicated what the work program looks like. It gets bigger and bigger every year with new projects taken on board. Every project has a generally 3 to 5 year life cycle, depending on how complex it is. So at some point in time, there might be a need for the program to think about making decisions around resource allocation and priority setting, just to ensure that we're in a position to put our efforts where the most benefit could be derived. Not sure we're at that point just yet, but certainly as every year progresses, it becomes more evident that there are finite resources amongst all of the OECD member countries to move some of these projects forward. And we see that manifest from time to time in projects, for example, switching lead countries, or some projects being placed in the annex because there are resources that were apparent at the beginning of a project, unfortunately for a variety of reasons, may not have followed through. We also have an ongoing challenge associated with the balance between making sure that the methods that are incorporated into test guidelines are supported by transparency. So there's obviously a need to understand how they work at a fairly granular level, because they support decision-making by countries that are making regulatory decisions on the basis of these methodologies. At the same time, incentivizing innovation. So if the program goes too far in one direction, it may de-incentivize those from developing methods because they may not be able to see a return on their investment. So this is a struggle and a constant balancing act that the program is going to, has been and continues to work through. Anne mentioned briefly the defined approach-based test guidelines. Essentially, without describing what those are in depth, just essentially methods that will allow the use of multiple methods together to address a particular endpoint or area where no single alternative method, in vitro method, in silico method may ultimately replace an animal test guideline. And these defined approach-based test guidelines use fixed data interpretation procedures to allow the grouping of multiple methods, such that it ultimately could replace an animal method and therefore be covered under the mutual acceptance of data principle. And so we were successful in April of this past year in approving the first defined approach-based test guideline for skin sensitization. And we would expect that this approach will continue in the future to cover other endpoints that are in this situation. We, as time has progressed, have developed a large number of test guidelines. There's a fairly significant library, and as time progresses, like in any area, you need to curate your library to make sure it's most relevant and current. And so as methods become more obsolete or just advance in age and need revisions, or where there might be animal methods that are used that may not be, or incorporated that may not necessarily be widely used, we do have the need to start to consider how we can address those. And there's been some work recently dedicated to addressing the curation of the program, and some work by countries to help to set priorities for how that could come together. And then lastly, preserving mutual acceptance of data in light of different requirements in different jurisdictions. Obviously, where requirements are unique to a particular country or region, it's always tempting to go off on your own, but the value of the multilateral approach has been well established. And so the efforts that various member countries play in coming to the table and ensuring that we're able to set test guidelines together that have broad applicability is going to become and continue to be very important. And so then in conclusion, my last slide here, just to indicate that my view, of course, is that the program is really well positioned to translate advances in science and method development to test guidelines, as it always has been, and it will continue to be over the foreseeable future. And it's supported as well by an enormous community of experts, as Anne indicated, that contribute to the program development. And that provides a really solid foundation for the future. And then, of course, the support and advanced mutual acceptance of data couldn't happen without extensive collaboration with the Working Party on GLP. And so we would definitely welcome that going forward. So thank you for your time today.
Now we are going to move to our next speaker, Valérie Zhuang, yes. From the European Union Reference Laboratory, EUREL-ECVAM, European Center for the Validation of Alternative Methods, who is going to give us the perspective of a validation body who does a lot of work, who contributes a lot for the program, like also many member countries, of course, but validation is certainly a central element of test guidelines development. So thank you, Valérie, for your presentation.
Morning, good afternoon, or good evening to everybody, depending on where you are in the world. My name is Valérie Zhuang, and as Anne already and Eva already introduced me, I'm the National Coordinator for the European Union. And I'm working at the European Commission, more precisely in the Joint Research Center of the European Commission, which I will introduce in a moment. My presentation will focus on validation in a regulatory context. And here's an overview of my presentation. First, I will briefly introduce the JRC and UL-ECVAM. Then we will go through the definition and principles of validation, novel concepts in the validation of alternative methods, and finally to the lessons learned during validation studies with regard to scientific and non-scientific issues. The Joint Research Center is the science and knowledge service of the European Commission. It supports EU policies with scientific and technical evidence that is independent from any national or sectorial interests. It has about 3,000 staff, almost 70% of whom are scientists. The headquarters are located in Brussels, and the research facilities are spread over 5 EU member states, which you see here on this map, namely ISPRA in Italy, Karlsruhe in Germany, Gale in Belgium, Patton in the Netherlands, and Sevilla in Spain. The largest site is at ISPRA, and that's also where our unit is located. Our unit is part of Directorate F, which is on health, consumers, and reference materials, and is called Chemical Safety and Alternative Methods Unit. The unit hosts UL-ECVAM, the European Union Reference Laboratory for Alternatives to Animal Testing, that has been established under Directive 2010/63 on the protection of animals used for scientific purposes, with the following broad tasks and duties: guide research on alternative methods, coordinate validation within the EU, disseminate information on the trias, promote stakeholder dialogue, and promote international acceptance. We have been working on method validation for a long time. In fact, we celebrate our 30th birthday this year. And over those years, we estimate that we have dealt with well over 100 methods with a view to their validation and regulatory use. We are always looking forward to share our knowledge and experience and to discuss ways of evolving validation principles and practice to keep pace with scientific and regulatory developments and to be as efficient and effective in what we do. We work on test methods or approaches throughout their whole life cycle, from research and development over validation to international adoption, regulatory acceptance, and finally implementation. We don't do this in isolation, but we have set up various networks that provide input to our process. For example, PARERE, our network of regulators from EU member states, relevant Commission policy DGs, and EU agencies such as ECHA, EFSA, and EMA. They provide a preliminary assessment of regulatory relevance of a proposed test method or approach, whereas our ECVAM stakeholder forum, called ESTAF, composed of industry, academia, and civil society, provides advice on the relevance of a method or an approach from a user perspective. The international perspective is provided by ICATM, the International Cooperation on Alternative Test Methods, involving NICETM ICVAM in the US, JACVAM in Japan, Health Canada, COCVAM in South Korea, BRACVAM in Brazil, and occasionally governmental institutions from China, Singapore, and Taiwan. We have also set up and coordinate a network of 35 validation laboratories called EU NetVal, which provides support for prospective validation studies. Our ECVAM Scientific Advisory Committee, called ESAC, is carrying out independent scientific peer reviews of validation studies and issues opinions on the scientific validity of a test method or an approach. In certain cases, this opinion is followed up by the publication of a UL ECVAM recommendation that has undergone prior consultation by Parere, ESTAF, ICATM, public, and the test submitter in case the recommendation is based on a test method that has been submitted to ECVAM for evaluation, validation, and/or peer review. Finally, international adoption and regulatory use is achieved through uptake of the scientifically valid method into the EU test method regulation, or in an OECD test guideline, or as an ISO standard, or for medicinal products in ICH, VICH guidelines, or at the level of the pharmacopoeia, depending on the sector. Now we go to the definition and principles of validation. Validation principles of toxicity test procedures were first discussed at an CAT-ERGAT workshop in 1990. CAT stands for Center for Alternatives to Animal Testing, which is still existing and it's located at the Johns Hopkins University in the US. And ERGAT stands for European Research Group for Alternatives in Toxicity Testing. Then these principles were then taken up later in OECD Guidance Document 34, that was in 2005. And validation has been defined as the process by which the reliability and relevance of a particular approach, method, process, or assessment is established for a defined purpose. Reliability is the extent that a test method can be performed reproducibly, yes, within and between laboratories and over time when using the same protocol. Whereas relevance is the extent to which the test correctly measures or predicts the biological effect of interest. The defined purpose can be various and range from addressing a specific regulatory information requirement, for example, for classifying a chemical as a skin irritant, to the generation of relevant mechanistic information. I will come to this in a minute. A test method is never valid in an absolute sense, but only in relation to a defined purpose. If you are interested in the historical perspective on the development, validation, and use of alternative methods in toxicology, this book here is a good reference. Another useful reference is the book on validating alternative methods for toxicity testing. This book provides information on best practices and new thinking regarding the validation of alternative methods for toxicity testing. An in vitro test method is usually composed of a test system, endpoints or biomarkers, and a prediction model or data interpretation procedure, and is accompanied by a protocol that describes how you should perform the test. The test system could be cells or tissues, such as, for example, a reconstructed human epidermis model. The endpoints could be, for example, MTT reduction for measuring cell viability, and the biomarker could be, for example, LDH release or an expression of certain genes. The prediction model is a formula or an algorithm used to convert the results generated by a test method into a prediction of an effect of interest. And the data interpretation procedure is used to determine how well the results from the test predict or model the biological effect of interest. Both test system and endpoint need to be reproducible and have a relevant scientific basis, whilst the prediction model and data interpretation procedure need to have a predictive capacity. Regarding the purpose, as I said before, you could use the test methods for various applications in regulatory decision-making. For example, to obtain mechanistic information, to investigate how a chemical interacts with a human protein, or for priority setting of chemicals of concern that need to be further tested, for hazard identification, for classification and labeling purposes, or for hazard characterization with a dose response assessment and the establishment of a threshold dose below which the toxic effects are no longer observed. Considering what was just said, validation can thus be seen as the ultimate means to build confidence and reach acceptance, but also to ensure that solid science-based decisions are made to protect human health and the environment. Validation, as we have heard before as well, facilitates the development of globally harmonized standard methods. And as such, it should not be perceived as an impediment or constraint. Acceptance cannot simply be imposed, but needs a process of trust building amongst various actors. In 2004, ECFAM proposed to break down the information required for peer review of a test method into 7 modules that do not necessarily need to be assessed in a sequential order. And this provides a certain flexibility. However, test definition would normally always come first, but could still be refined at a later stage in the process. The 7 modules that you see here on this slide are test definition, within laboratory reproducibility, transferability, between laboratory reproducibility, predictive capacity, applicability domain, and if appropriate and necessary, performance standards. Within laboratory reproducibility, transferability and between laboratory reproducibility relate to the reliability of a test method, whereas predictive capacity and applicability domain relate to its relevance. At the level of the first module, namely the test definition, you normally already have an idea and at least some preliminary data on the reproducibility and relevance of a method. Now, this slide is just to show that there is not always a need to carry out a full prospective multi-laboratory validation study, but that you could also validate a method on the basis of existing data in a retrospective validation study, or for similar methods on the basis of performance standards, if these are available. And on the right-hand side, you can see several examples of methods in OECD test guidelines that underwent different types of validation processes. Now to the novel concepts in the validation of alternative methods. Now, nowadays, we are faced with a plethora of new technologies, such as cheminformatics and computational models, organ-on-a-chip devices, high-content imaging, omics technology, 3D tissues, exposure modeling. All the data generated is then integrated to predict safety. The biological complexity of in vitro test systems is increasing. We are speaking of cultures of multiple cell types grown together. Furthermore, we are experiencing a convergence of different scientific disciplines, such as biology and engineering, physics, electronics, and so on. So validating or standardizing an approach starts to have a broader meaning depending on what it refers to. Thus, the principles of validation that say that a method or approach should be relevant and reliable are universal and valid, the validation process is adaptable and depends on the purpose. One size does not fit all. The process needs to be fit for purpose. I now come to the lessons learned from carrying out validation studies. Several workshops on validation had been organized in 2017 and 2018. In 2017, IVM and BFR, so the Netherlands and Germany, organized a joint workshop where the drivers and barriers concerning implementation of innovative 3Rs approaches in regulatory toxicology were discussed. This was followed up by a second workshop in 2018, aiming to develop feasible solutions and recommendations to move innovation forward in this area. In 2018, we organized a workshop with our ICTEM partners and invited also some additional governmental institutes and authorities. The OECD participated as well. At the ICTEM workshop, we discussed the lessons learned by ICTEM partners in the conduct of validation studies. Then we discussed 4 challenging topics, in particular, how to establish relevance without animal data, how to evaluate reliability without ring trials, how to evolve validation practices, and how to develop the concept of standards. For the lessons learned, here are some scientific issues we came across. How should the relevance of single methods be assessed if you know that a method needs to be integrated with several others? Relevance assessment should be made within that integration context, and one should not emphasize on the relevance of the individual method. Single methods validation is also time consuming and costly, in particular if methods are not meant to be used individually. Another issue that has been encountered is the poor or unknown quality of the reference data, which are often in vivo animal data, against which you validate your in vitro data. And related to this, too high focus is given to the animal test as the gold standard and the derived data which are used to assess the predictive capacity of your test, whereas animal data are not always human relevant. Another issue is poor chemical selection, introducing bias. For example, when the same chemicals are used for the development and validation of the test method, and sometimes the same chemicals are even used for transferability and training purposes. Further guidance on chemical selection is needed to better explain how this should be properly done. Lab-specific SOPs that hinder transferability. For example, the SOPs are too specific to one laboratory, too detailed with regard to the material that should be used, and this is hindering transferability. Regarding non-scientific issues, the current regulatory requirements are based on animal studies, whereas the new non-animal methods provide a different type of information that does not fit these requirements. International harmonization is critical, but having guidelines does not guarantee use by regulators and industry if they do not fit the regulatory requirement in a jurisdiction. Engagement with stakeholders at every step is important. A review and evaluation of uses and needs of data across end users is key to define fit for purpose and context of use. We heard about intellectual property, tackling intellectual property, ensuring commercial availability and avoiding abuse of monopolies. This is a topic that was discussed considerably over the recent years at the OECD with the development of the guiding principles on good practices for the availability, distribution of protected elements in OECD test guidelines, and the implementation of fair, reasonable, and non-discriminatory which we call FRIEND, licensing terms and conditions. Lack of transparency of protected elements, for example, when a test method includes confidential business information and the prediction model is not explicitly described. GLP implementation of protected elements and cloud-based computerized system. That's also a topic that has been discussed more recently in the OECD Working Party on GLP. So some final thoughts from my side. Validation will remain one of the cornerstones of a successful toxicological evolution or revolution. Validation needs to keep pace with rapid scientific progress. For example, the defined approaches, computational models, organ on a chip. It is important to keep scientific integrity, credibility, and usefulness while making the process more efficient. And validation should be framed as a process to characterize and reduce uncertainty rather than a ring trial to demonstrate toxicological equivalence. That's all from me. Thank you very much.